Hi, we'll continue talking about transcription and translation. As a component of the process of translation, the folded structure of transfer RNA promotes its decoding functions. Transfer RNA are 70 to 80 nucleotide long. In bacterial cells, 30 to 40, there are 30 to 40 transfer RNAs, while in animal cells, there are 50 to 100 transfer RNAs. The structure of transfer RNA in solution fold into stem loop arrangement that resemble a clover leaf when drawn in 2 dimensions. It consists of 4 base paired stems and the 3 loops. You can see that the 3' end of the unlooped acceptor stem always have a specific amino acid where the specific amino acid is attached, always have the sequence of CCA, as you can see from the figure. Several bases in most transfer RNA are modified after transcription, and they play an important role in protein synthesis. The transfer RNA can be viewed in 3 dimensions as the folded shape in the form of L-shaped. The amino acids become activated when covalently linked to transfer RNA. To participate in protein synthesis, a transfer RNA must become chemically linked to specific amino acids. This linkage is catalyzed by a specific set and a battery of enzymes called amino acid transfer RNA synthase, and there are 20 different ones of them. The amino acid is linked to transfer RNA by high energy bond, and hence the transfer RNA is activated transfer RNA. The energy of this bond is used for the formation of the peptide bond linking adjacent amino acid in a growing polypeptide chain. Because some amino acids are so similar in structure, amino acid transfer RNA synthetases sometimes make mistakes. Amino acids become activated when covalently linked to transfer RNA. These amino acid transfer RNA synthetases have a proofreading characterization in which mistakes are corrected by the enzyme itself. The error rate for translation in E. coli is very low, 1 in 5,000 codons. Here you can see the amino acid transfer RNA synthetase specific for phenylalanine. The amino acid attached to the enzyme, which is phenylalanine, and then the transfer RNA specific for the phenylalanine attached to the bind to the enzyme. Then a high energy bond is formed between the transfer RNA and the amino acids on the expense of energy, ATP. Now the net result, phenylalanine, is selected by its codon because the transfer RNA will recruit the amino acid to the codon on the messenger RNA. Messenger RNA carries information from DNA in a 3-letter genetic code. Messenger RNA can have 64 possible codons, 4 by 4 by 4, which is a universal code that specifies amino acids. 61 specify amino acids, while 3 stop codons do not specify amino acids. Methionine and tryptophan have only 1 single codon, while leucine, serine, and arginine have 6 different codons. The synthesis of any protein in prokaryotes and eukaryotes begins with methionine, where the start codon is AUG. Sometimes GUG is used as the initiator codon in a few bacterial messenger RNA, while CUG rarely is used as an initiator codon in eukaryotes. The reading frame actually it is the sequence of codon from a specific start site to a stop codon. Theoretically, a messenger RNA can be translated into 3 different frames, reading frames, as you can see from the figure, frame 1, 2, 3. The difference actually when the translational machinery skips 1 nucleotide at the beginning and starts the codes after that. So you can see this in frame 2 skipping 1 nucleotide and frame 3 skipping 2 nucleotides. But most messenger RNA can be read only in 1 frame, because the stop codons encounters in the other 2 possible reading frames and terminate the sequence before a functional protein is produced. Here you can see the table that dictate the codes dictates the codes for the amino acid. So you can see the first column is represent the first position of the codon. These four columns represent the second position, while the third one represent the third position, the last column. So for example, if you have these three codons, the first codon is GCU, and keep in mind that reading starts from the 5' end of the messenger RNA. So the first codon is GCU, and this will be translated into alanine. Same alanine. Same you can try for the other two codons. You'll see that it is translated into cysteine and leucine. Ribosomes are protein synthesis machines. Ribosomes are the most abundant RNA complex, protein complex in the cell. All species have structure and function similarities reflecting common evolutionary origin. So the structure have been determined of the ribosomes. The structure have been determined by cryo-electron microscope. Ribosomes direct elongation of a polypeptide at a rate of 3 to 5 amino acids per second. That's mean average 4 amino acids per second. Small protein, 100 to 200 amino acids, are made in minutes or a minute or less. Titin is the largest known protein found in muscles. It takes up to 3 hours to be made, 30,000 amino acids. A ribosome is composed of different ribosomal RNA and proteins. A ribosome is organized into large and small subunits. Here you can see the ribosome component. In E. coli, it consists of 3 ribosomal RNAs and 54 proteins, while in S. cerevisiae yeast, 79 proteins and 4 ribosomal RNA, while in human 80 proteins and 4 ribosomal RNA. For the large subunit, you can see that there are 33 proteins, and these are the types of RNAs, and so on for yeast and human. For the small subunit, you can see the difference between the three. So what's the Svetric unit S? Svetric unit is a measure of the sedimentation rate of the suspended particles of the protein of the small and large subunits under standard condition. And actually it's named based on the scientist who put the the nomenclature for that is Svetric. Here you can see the structure of ribosome with the larger and the small ribosomal subunits while binding to the messenger RNA. You can see that the transfer RNA loaded with amino acids is arriving to attach to the complementary codon, and then the peptide bond is formed between the incoming amino acid with the growing polypeptide chain, and once the transfer RNA unloads its amino acids or the polypeptide chain, it exits from the structure. Here you can see that the ribosomes generally have 3 sites, A and P and E site. For the A site, it's the amino acylated transfer RNA where the amino acylated transfer RNA bind, the incoming transfer RNA loaded with amino acid. For the P site, actually in the P site, transfer RNA linked to the growing polypeptide chain, the linkage occur here in that site. While the E site, it occupies the transfer RNA that that already had unloaded the amino acid it recruits. Both prokaryotes and eukaryotes contain 2 different transfer RNA for methionine, so it's a specific feature for the transfer RNAs of methionine. The transfer RNA I MET can initiate protein synthesis at the B site of the ribosome, while transfer RNA MET can add methionine into growing polypeptide chain and enters the ribosome at the A site. So the peculiar one is the transfer RNA I, which ensures that the methionine is the only amino acid that will be loaded to the ribosome at the initiation codon, while the other transfer RNA is used to add the methionine whenever the codon for which is A or G appears in the messenger RNA in any sequence. The same amino acid transfer RNA charges both transfer RNAs with methionine, so both of them use amino acid transfer RNA synthetase. AUG codon is the start codon in messenger RNA, is only recognized by methionine transfer RNA-I. Now we'll talk about the stepwise synthesis of proteins on ribosomes. Like transcription, translation consists of three stages: initiation, elongation, and termination. We'll start with elongation. It starts with the small ribosomal subunits bound to the eukaryotic initiation factor 3. With the addition of eukaryotic initiation factor 1a and eukaryotic initiation factor 2 bound to GTP plus the transfer RNAi, which will form a tertiary complex which is called pre-initiation complex. And note that the transfer RNAi loaded with the methionine is bound to the P site of the small ribosomal subunit. Here the messenger RNA are ready to bound to this subunit of the ribosome. The messenger RNA is bound to EIF4, the cap binding complex, and together with the pre-initiation complex will form the initiation complex. This messenger RNA looks like a tertiary circular complex, and you can see that by the helicase activity of 4A eukaryotic initiation factor 4A, which have helicase activity, will make sure that the messenger RNA is not into the structure of a secondary or tertiary structure. There is no stems and loop, because messenger RNA needs to be linear to be easily translated. Soon after that, the initiation complex start to scan for the EUG in a sequence called the Kozak sequence. Once the recognition is done to the Kozak sequence, this recognition hydrolyzes GTP that is bound to the transfer RNAi and prevent further scanning. So it's an irreversible step to ensure that the translation continues. Soon after that, all of these factors is released, and now the ribosome is ready for continuing translation and accepting the large ribosomal subunit, as you can see. Now the ribosome is termed ATS ribosome. For the elongation step, you can see that now the ribosome is ready to accept a new transfer RNA at the A site. So you can see a successive transfer RNA loading with the elongation factor 1 alpha is recruited with amino acid 2, 3, and 4. If the anticodon base pair with the codon, the GTP is hydrolyzed, releasing the EF1 alpha. So you can see now the amino acid 2 recruited by the transfer RNA is located on the A site. Here, by the attachment releases EF1 alpha by the hydrolysis, leading to a conformational change which enables the transfer RNA to be near the first transfer RNA loading the amino acid number 1. So you can see that the peptide bond is formed between the two amino acids 1 and 2. Soon after that, the ribosomes moves along the messenger RNA. So now is the first transfer RNA is shifted from the P site into the E site, and it is empty, so it shifted to the exit site. And now the A site is free for accepting a new transfer RNA. And so on, and you can see that the step is repeated again until all the amino acids, all the messenger RNA is read. For the termination, once the stop codon have been reached, which is in this case is UAA, a release factor, a eukaryotic release factor 3 bound to GTP, which is similar to the release factor 1 bound to release factor 3, similar to the shape of transfer RNA, will block the process of translation. This leads to the release of the all component, and you have now the nascent polypeptide chains and the unactivated transfer RNA. Soon after that, the nascent polypeptide chain falls into 3D conformation by the help of group proteins comes from the endoplasmic reticulum. These group proteins are called chaperones. So actually, a multiple number of ribosomes can attach to the same messenger RNA to increase the efficiency of translation. So in a process of recycling of ribosomes. So actually two phenomena significantly increase the overall rate of protein synthesis. The first one is the simultaneous translation of a single messenger RNA molecule by multiple ribosomes, and as well the rapid recycling of ribosome subunits after they disengage from the stop codon. This structure of the binding of a number of ribosomes to the messenger RNA is called polyribosomes or simply polysomes. So you can see here that the ribosomes start with a small ribosomal subunit at the 5' end. Then soon after that, the ribosome shifts and moves along the messenger RNA. Once the ribosomes moves from the start initiation codon, another ribosome starts to do translation until reaching the single ribosome reaches the stop codon UEG. It is released and recycled and can engage again from the initiator codon to start a new translation round. And as I said, this structure is called the polyribosomes. So we'll talk now about the post-translational modification. Actually, PTM is a covalent modification on proteins that occur after DNA has been transcribed into RNA and translated into proteins. The major types of protein PTMs, actually post-translational modification, is phosphorylation, acetylation, glycosylation, methylation, hydroxylation, and you can see from the figure a lot more others PTMs. So actually we'll talk about three types of this PTM is the hydroxylation of the Pro-alpha chain and the formation of collagen, the histone tails modifications, and the protein degradation. We'll take the first modification is the ubiquitin and the proteasome-mediated proteolysis. So you can see here during the process of ubiquitination, which finally leads to polyubiquitination and then proteolysis mediated by the proteasome, it starts with the ubiquitin activating enzyme that adds this ubiquitin activating enzyme. Actually, it is a small polypeptide of about 76 amino acids that is added on the lysine residue of a protein, and after that there is a subsequent addition of more ubiquitin molecule to the same residue, leading to the polyubiquitination of protein. This ubiquitin needs energy in the form of ATP, and the E1, the ubiquitin activating enzyme, after that the engagement of E2 and the release of E1, the E2 is a ubiquitin conjugating enzyme. After that, the protein, the target protein, need to be degraded by the help of E3 that is recruited. This ubiquitin is added on the lysine, specific lysine on the target protein. After that, the steps are repeated for n times until polyubiquitination occurs on the same lysine for the target protein. This polyubiquitinated protein will be recognized by a proteasome, which will tend to cleave the protein into small peptide chains. So you can see here, this is the structure of the proteasome. It's a machinery inside the cytoplasmic, one of the cytoplasmic machinery inside the cytoplasm that looks like a barrel shaped, which have one end that accept the polyubiquitinated protein, and soon, before entering to the proteasome, the ubiquitin is released in a process called deubiquitination to be recycled, and the protein is internalized into the proteasome as a primary structure, and soon it released out as a peptide. Of course, this process expends energy in the form of ATP. We'll talk here about the hydroxylation of pro-alpha chain as a step in the formation of collagen fiber and as a second example of post-translational modification. You can see initially that the alpha chain are translated by the ribosomes in the rough endoplasmic reticulum lumen. With the help of vitamin C and the process of hydroxylation occur on the polypeptide chain, actually on the majorly on the amino acid proline. That's why it's a hydroxylation of pro-alpha chain. Once it's hydroxylized, it forms with the aid of the hedgehog protein 47 HSP47. You have the pro-collagen structure. This pro-collagen structure is translocated into the Golgi apparatus for lateral association. Soon after that, it is released into the extracellular space where a cleavage occurs for this propeptide to form the collagen molecules. These collagen molecules, you can see when you enlarge it and see it under the electron microscope, you can see the fibril of each of these collagen fibers. This fibril polymerized together to form a collagen fiber that soon form the vesicle that soon form the tendon. Finally, the collagen is formed. The glycosylation is an example as well of post-translational modification, and this process of glycosylation actually involves a lot of modification on the surface, on the cell surface. So you can see initially the ribosome translate the protein of interest where a molecule of carbohydrates is being attached to the translated polypeptide chain. Soon after that, these polypeptide chains undergo some modifications until released from the Golgi apparatus to be expressed on the surface of the cell membrane of the cells. The next example of post-translational modification is the modification of histone tails. And actually this is an important modification because it controls the chromatin condensation and function. You can see each of the histones protein making up the nucleosome core contain histone tails, as you can see from the figure, extending from the globular histone octamer core. The histone tails are required for chromatin to condense from the beads on string chrome formation into the 30 nanometer fiber. Histone tails are subject to multiple post-translational modification. The particular combination of the post-translation modification is called histone codes. So histone codes is important to control if the gene will be exposed for transcription or not exposed, if the chromatin is condensed or not condensed. The influences that in this histone code influences chromatin function by creating or removing binding site from chromatin associating proteins, especially like the RNA polymerase. So you can see here that the modification you can see on the histone tail occur acetylated lysine can be acetylated on the position histone 3, K9, K14, K18, K27, K56, and there is a lot of examples for acetylated lysine, specific lysines on histone 4, 2A and 2B, and all these signal leads to activation. So generally acetylated lysine leads to activation of genes of transcription, while hypoacylated lysine leads to repression. You can see as well phosphorylated serine or threonine at this position leads to activation of transcription, while methylated arginine at this position leads to activation, and you can see as well there is methylated lysine at a different position. Some of them will lead to activation, others will lead to elongation, others will lead to repression. Other ubiquitinated lysine will lead as well, one of them will lead to activation, other one will lead to repression. So the DNA compaction can be observed by light and electron microscopy using dyes that bind DNA. condensed region of red chromatin termed the heterochromatin, the dark region in the figure, while the less condensed chromatin is the euchromatin, is the light. So the heterochromatin include the centromeres, telomeres, and inactive genes. So you can see in heterochromatin, actually it is the inactive condensed form. The H3 had to be look like the methylation of trimethylation of lysine 9 and the trimethylation of lysine 27. While when it's active or open in the euchromatin, you can see there's a different modifications on different amino acids, either acetylation, phosphorylation, or trimethylation, and this termed the histone code. So basically, this part of the gene that is wrapped by histone 3 had to have this specific combination of modification to open and inactivate transcription of that gene. So that's why it's called histone code. So you can see here for all the histone 1, histone 2A, 2B, and 3 and 4, there is a multiple combination of modification you can see. All of them with a different combination can lead to activation or inhibition of a certain genes. Keep in mind that a woman who smokes while pregnant will induce an epigenetic changes in 3 generations at once in herself, her unborn daughter, and her daughter's reproductive cells. Actually, why it's we are talking about epigenetics? Because histone modification is a type of epigenetic modification, because it actually not affecting it is affecting the gene by not affecting the structure of genes, not by doing mutations, but by including a signal that will affect the behavior of the expression of the gene, either activation or repression. So that's why it's an epigenetic mechanism. So why post-translational modification is important? Actually, in other words, why or how there is a protein complexity or the proteome complexity. Keeping in mind that the number of protein coding genes in the genome is inadequate for explaining the differences in biological complexity. So several phenomena can generate more complexity in the expressed protein than in predicted from their genome. First, the process of alternative splicing of pre-messenger RNA can yield multiple functional messenger RNA in human. The mean number of alternatively spliced messenger RNA expressed per gene is about 6. So actually a gene can generate 6 different proteins from the same genes based on the alternative splicing, while the second level of the complexity of the proteome comes from the variation in the post-translational modification of many proteins that produce different functional differences. So you can see the same protein, when it's methylated, it will do a certain job, and when it's phosphorated, it will do another job. When it's acetylated, it will do a third job. This gives it functional difference due to the post-translational modification. Thank you.